I'm very excited to speak to you all today. We have, obviously we could talk a lot about Apex. There are so many fun functionalities, but today we're gonna have a very specific focus, and that's gonna be on the curation of data with Apex and, being as efficient as we can with gathering data and making the summary tables out of your results. So the agenda for today is, it's really like an interactive workshop. We will take questions at the very end, but feel free to write in the chat, and Baron and Jason can maybe answer some of the questions as we go along. But the plan for today is really to go over how you can set up plate layouts. So using the layout function in the Apex, so labeling your wells with information such as donor information, stimulus information, replicate information, and so on. We'll also show you how to use the template function so that you don't need to, adjust settings and relabel all of your wells every time you run a new plate from the same type of experiment. So this step one is really going to be linked with making your data acquisition faster. We're also going to look at how to export data from multiple plates into a single Excel. So if you are working with several plates at a time or over several months or sometimes even years, but you're using the same settings, the same kind of layouts, what you can do is to export your spot counts or spot volume values into a single Excel and make a single summary data table. So this is what we're going to go into, and we'll show you also how to do that with the pivot table function in Excel. And as I said, the last part of the seminar or the webinar is really to answer any of your questions that you may have. So I just want to give you an outline on the plates and the data that we will work with today. And our R&D scientists have kindly prepared several plates for us to work with today. So we have 2 ELISpot plates. They are interferon gamma ELISpot plates developed with ALP. And we have 4 donors that we have stimulated with different stimuli. So we have, on the plate 1, we have 2 donors that have been stimulated with SARS-CoV-2, CMV, and PHA. And we also have the unstimulated control, which includes basically cells without any stimulus, and the media control, which is just the media, no cells added. You can also see how many cells per well were added in each of these wells. So in the media control, again, there's no cells added. In the unstimulated SARS-CoV-2 and CMV wells, we have 250,000, and in the PHA, we added 50,000 cells. So this is plate 1, and in the plate 2, it has exactly the same layout in terms of stimuli and cells per well, but we have donor 3 and donor 4 instead. So I would like to start with this part, and I will open Apex. And as Jason mentioned, your Apex may look a little different depending on which version you have. I am working with the Apex 2.0. And if you want to upgrade to this software version, you can just reach out to Jason and he can help you get sorted. But I will start diving in right into the action. And we're going to start with our ELISpot plates. So I have 2 plates that are white plastic. Maybe you can see it through the camera. And I'm going to use the MSIP white assay principle, double click on that. And in this plate, I didn't have the full plate that I have used. So I've only used these wells. So I'm just going to highlight them and then just press read. And of course, you are all users of our instruments, so you know how quick and easy it is to get this done. And as I'm reading, I can already, start playing around with data. I could adjust brightness and contrast by going into the presets here. So I can make it a little brighter if I want to, maybe a little less contrast. And it's interactive while you're reading. So I'm just going to finish this read. And I can go into a single one of the wells, let's say, and inspect how the spots are being counted. So obviously, if this is the first time you're doing this experiment, you would be then deciding on your size and intensity parameters. We won't go into too much detail today about that part. But this is something that you need to decide for every single experiment that you have, and then stick to those settings for the rest of your plates. But for this purpose, I might just set it a little darker and a little more contrast. Now, brightness and contrast don't change the spot counts, while the size and intensity does. So maybe in this case, you want to focus on a bit larger spot. So I'm just going to increase the size threshold a tiny bit, maybe to let's say to 180. And maybe if you are happy with these settings, then we can keep those settings and not change them anymore. As you can see, they have been updated on the preset here. And now, of course, all we have is the spot counts, but we don't really have information about what has been added where. So we can do that. And there are multiple ways of doing it. If you have Apex 1, you probably are familiar with manual labeling function and adding the layouts manually. So one way to do it would be to highlight the wells that you want to label with a specific label. So let's say these ones. And then right-click, go into the layouts, in the donor, and add label. So you can see that now a new label appears in green. And if I go under the donor section here, you'll see that that new label is added. Now I can rename that to, let's say, donor 1. And you can see that that's being updated in real time. So while you can do it like that manually for everything, for donor, stimuli, replicates, cells per well, it's quite laborious and it takes a lot of time. So we have built in a faster feature to help you label your wells quicker with this information. And I hope you can see it on your screens, but there are these two little hidden buttons next to the layout. So if I press on one of them, which is export layout, and I'm just gonna save that Excel file, I'll show you what that Excel file has. Let me open it. Here, so this is basically a template that you can use and fill in and later import into Apex. So right now you see that I have already added donor 1 from before, and I can continue filling up these wells or these cells in the Excel with the information I want to import. And then I can show you that I already have pre-labeled this in another Excel file. So I'll just show you how to do that. If you go back to the plate and Apex, there is this button here for import layout. I'm just going to press on that, choose the plate layout that I've had created. And now you can see that a bunch of labels have been added here. So if I highlight the two donors, as you see, the donor 1 is here, but I've also added donor 2, cells per well information, the different stimuli information, as well as the replica information. So this is a really much faster way of adding the layouts. And in the Apex 2, you can do it with Excel. So that's just what I want to show you, that there is a faster way to do this than doing it manually. And so let's say that you have now ran these two donors, but you really would like to repeat the experiment with 2 other donors, but you have to do it another day or on another plate. And of course, you could just create a new plate, create a new plate and do everything from scratch. You could go and add in these settings manually. You could add in the labels, but there is a faster way of doing it, which would be to create a template. And to create a template is very easy. So now we are in the space where we have, decided on the settings and the labels. What we can do is go under the save button here. We press these 3 dots, and then there is a function to create a template. So I'm going to click on create template, and I can say ELISpot template 1, and I'm going to save it on my desktop. So now this template is exported. And when I go into the new plate, you're of course familiar with the assay principle tab. So that's when you are taking a plate from scratch. But there's also a plate template tab. So if I go into the plate template tab, I can add a template. And then I'm going to go into the desktop, find this ELISpot template file, which I usually call ZMT, and then just going to press open. So you see how I have imported this template now into Apex, and I can double click on this. And now, if you remember, the settings that I have used have stayed the same, and the labels have also stayed the same. So we have already pre-labeled the stimuli, we have already pre-labeled how many cells were added and which donors there were. So now what I'm going to do is I'm going to read the new plate that I have with me. So that would be your second plate on your second day experiment. I'm going to insert it. And now I can just highlight these wells and read the second plate. So of course now, because I'm reading the second plate, these donors are no longer the same. So all I need to do is to really just change the label into the donor names that are representative of what I've added. So this is going to be donor 3, and this is donor 4. And I can do that while the plate is reading. And thankfully, ELISpot readout and single FluoroSpot readout is very quick, so it doesn't take very long. So now we have read basically 2 different plates, and I'm just going to save them just so that we have them. So I'm going to call this plate 1. I'm going to save it on the desktop for easy access. And then with this one, we have a plate 2. So I'm also going to save this one. And of course, one way to analyze the data, get the spot counts from it, would be to go into the individual plates. So let me just show you that method as well. So if you go into the plate 1 folder, this is the data output that you get. And this is the Excel that comes with the plate 1. So if I open this plate 1, you'll see that we have the classical readout of, of the plate layout and the spot forming units, the RSV, some RSV, information about what has been added, and so on. So that's plate 1. Now, plate 2 information is, of course, on the folder from plate 2. And that's here. And of course, you can copy and paste information from all of these plate folders into your own Excel. But we do have a faster way of doing this. And so I really want to show you this method because it can really save you a lot of time. And when I actually learned about this, I could open up to, I think I had about 15 plates or so that I have opened in Apex and were able to export data from all of those 15 plates into a single Excel. So how do we do that? So now we have plate 1 open and plate 2 open. All you need to do is go under Export, choose All Open ELISpot Plate Excel, and save this on your desktop, let's say. So now we've exported this Excel. I'm going to open it, and I'm going to show you what the output looks like. And I hope you're not too surprised by how much information there is here. But basically, this is a plate database for both plate 1 and plate 2. And if you look at the information, the first column here is indicating what plate you're looking at. So you can see that all of this information is coming from plate 1. And if I scroll down, you will start seeing plate 2 appearing. So that means that now we have a single Excel with both plate information exported in a single tab. And this is where we're going to go into the Excel pivot table function, which I hope once you learn this kind of analysis, it will really save you time. So the way that you can go into the pivot table analysis is that you can highlight the full table here. All you need to do is press Control A. With Control A, it's just going to automatically highlight the full table, all of the information in this tab. And then we're going to go into the Insert, and then we're going to insert pivot table. So we're going to choose new worksheet so that we create a new tab. And here we are. So pivot table, all the information that was in that massive data table is here. These are the columns of that table. So let me just go in between those 2 tabs. So the plate database, if you remember, had plate, well information, when the plate was read, the spot forming unit data, average RSV, all of that. So all of these column names are now becoming variables here. So all we need to do is really to drag and drop information that we're interested in. So let's say that you want to add your donors as your columns. So we have a layout function here. We can just take that layout donor and drag it onto the columns. And you can see now that donor information is appearing here. So we had 4 donors, right, from both plates, donor 1, 2, 3, and 4. Then let's say we want to look at the stimuli, and the stimuli could potentially then go into the rows, let's say. So the information about the stimuli are appearing as well. We have media, unstim, SARS-CoV-2, CMV, and PHA. And also maybe we want to plot our replicates for each donor, because if you remember, each stimulation condition had 3 replicates. So maybe we also want to put that information there. So we're going to take the replicate information from the layout and put it under the donor. So now you see each donor has 3 replicates under them. I haven't added any data yet. I'm just kind of building the outline of the data table. And I want to stress that this is very flexible. You can always switch donor with the stimuli information and so on. So there's a lot of flexibility when it comes to pivot tables. But now we kind of build the backbone. We also want to clean up a little bit these grand totals and subtotals because they don't really make that much sense. So I'm going to go into the design on the top here and choose do not show subtotals and no grand totals. So you can see it's a bit cleaner now. And also if you do have a few blank wells, so in our plate, we had all of this space here that was unoccupied. So that usually appears as blank in the pivot table. And I can zoom in a little so you can see better. So, but you can click on this button here and untick blank, and then the blank will also disappear. So now we've built the sort of the framework for the table. So if you are interested in spot forming units or spot numbers for all of these conditions, all we need to do is to take spot forming units and drag that under the values. Once I drag them into the values, we basically get all of these spot counts listed. And you have pretty much already made an aggregated data table that you can work with. Of course, there's so much more that you can do here. You could, for example, one thing and one trick I want to show you is how to get the average out of these 3 replicates shown instead of replicates separately. So now, to do that, you could just remove the replicates from here. And then in the values, if you just click under here, value field settings, you can choose average. And that's going to basically average those 3 replicates and give you an average number. So I'm just going to remove the decimal points. Oh, sometimes this happens that I don't get to remove them. Here you go. And now we have average spot numbers for each stimuli for each donor. And you can go further than that. You can, if you would like, you can insert a pivot chart, for example, so that can be a nice way to visualize your data. So now I'm showing for each donor, the responses and number of spots for each stimuli. So we can see that for donor 1, for example, that donor has a very, very strong CMV response, while donor 3 has highest PHA response and so on. So you can get a lot of quick information here. And yes, there's a lot more to pivot tables that we won't be necessarily going into today. But I just want to stress there's so much you can do. And you can really do some also interesting maths within the pivot tables. You can do normalization of data because we do have the information about the cells per well. So if I were to, for example, put that, I think, under stimuli, you would be able to see what kind of information, you know, how many spots you had or how many cells you've added and how many spots you get. So there's a lot of flexibility with pivot tables. Yes. So now I would also like, now we've all only covered the ELISpot analysis, but I would also like to show some FluoroSpot analysis. And I don't know if all of you are using only ELISpot, but even if you are only using ELISpot, it might be fun to see what the FluoroSpot data analysis would look like. And I just want to show you the difference in terms of data output that you get with FluoroSpot. So with FluoroSpot, it's really the biggest strength is that you get so much more data out of a single well compared to ELISpot. So if you look at, for example, in this triple color FluoroSpot, it's interferon gamma, IL-2, and TNF. And in this case, you could just be looking at each individual channel separately. So you're looking at interferon gamma total counts, IL-2 total counts, and also TNF total counts. But in addition to that, we have something even more exciting, which is you can look at co-secretory profiles. So you can get numbers of cells out of the well that are secreting 3, 2, or 1 cytokine at a time. So you can really get information from each well and each donor, each stimuli, what kind of response are the T cells eliciting. So you can have, in this case, we have 54 spots that are triple positive, 344 that are interferon gamma and TNF positive, and we have 298 that are TNF positive only and don't secrete interferon gamma or IL-2. So it's a lot more information, of course, but that also makes it really exciting. So I want to show you a FluoroSpot data. Let me just leave this. I want to show you a plate that I have pre-read, which had a very similar format to the plate 1 and plate 2 of ELISpot. It's the same donors, the same stimuli. It's just all done on a single plate. So I'm just going to open that plate quickly now. So here we are. And as I said, the layout is very similar. And I have already pre-labeled, or not pre-labeled, but I have created an Excel file that has the layout information. So I'm just going to import that. And so you can see that we have the 4 donors individually like that. The stimulation is the same like before. We have media, unstim, SARS-CoV-2, CMV, and PHA, and the same for the other 2 donors. Replicate information and also cells per well. So all of this information is there. But of course, now we're dealing with 3 different channels. So in contrast to ELISpot, where we're looking at one analyte at a time, which is interferon gamma in this case, we were looking at interferon gamma plates here. We're now looking at interferon gamma, granzyme B, and TNF. And so the way to preview a single color at a time, you can just untick here. And so in green, we have interferon gamma. So maybe you are aware already, but instead of having this called LED 490, you are able to rename this filter into an analyte name. I think that really helps later on with analysis when you're building the pivot tables. So the way to do that would be to go into the presets here, and you can press here and just rename this into interferon gamma. So I'm just going to do that. And you can see now that it's been updated to interferon gamma. You can do the same for granzyme B in this case. So I'm just renaming that to granzyme B. And lastly, we have TNF alpha. So now we know what we're looking at, which really helps when we look into a single well data. So now when I'm gonna go into a well view and look at this well, for example, you'll see that I'm getting a lot more information on this side. So we have information of the total counts, just like similar to ELISpot readout, where you're just looking at total response for each cytokine. So these are interferon gamma total, these are granzyme B total, and these are TNF. And then we have these co-secretory profiles. So we have some spots that are single positive for each analyte, double positive, and you can see them highlighted here in blue. And then the triple positive in this well it's actually just 1 cell. But so this one has secreted 3 analytes at a time. And you can always click on them and preview their intensity, size, and also relative spot volume values. Yes, so now that we have imported the layouts and have renamed the filters, we can basically just save this data. And I want to show you how the pivot tables look like in this plate. So let's just go into that folder and then open the FluoroSpot data. And of course, it's going to be a lot more data. So if you go into the spot forming units tab, you will see how many different information we get. So we get the total counts for LD490, LD550, LD640, and then the subset or secretory profiles counts as well. And it's quite overwhelming if you're doing this analysis manually by copy pasting information that you're interested in into another worksheet. So just like with ELISpot, where we use pivot functions to aggregate tables and make these summary tables, it can be done exactly the same way with the FluoroSpot assay as well. So if you look at the plate database, this is the enormous table that we can really use and simplify. So you can see how large this table is, but it basically has all the information about this plate. And because it has 3 channels, of course, it's extra data. But the way to analyze it would be very much similar. You press Control A again, so highlight the full table, and then we go into the Insert, Pivot Table, and New Worksheet, and we press OK. And I apologize if it's difficult to see these, but I am not able to zoom into these names, unfortunately. But I will try to zoom into the numbers when I move the information here. But so let's say we want to filter, or we want to make a similar table to before. So we again have layout donor information. We can drag that to the columns. Then we can take the stimuli information, put that into the rows. And then again, the same thing about cleaning up. That's just the Excel likes to show grand totals in pivot tables. And then we're going to remove the blanks. So we have a nice outline. Then we're going to move the spot forming units into our values and just choose that into the average so that it actually averages the replicates. And I'm going to clean up here. Let me just zoom in. I'm going to get rid of the decimal points. So you might think that, okay, now I have the information ready. This is my spot counts. But one thing to remember when working with FluoroSpot data is that, of course, we have 3 different channels. So we need to make sure that we are filtering on the channel that we're actually interested in. And this is where you can, this is easy to forget, but so it's always best to compare spot counts that you see here with the spot counts in the software itself. I think that's the easiest way to see if this makes sense. But yeah, just one thing to remember with FluoroSpot, that you need to filter on the channel that you're interested in. So we have the LED filter option here, and I can basically just drag it under filters. And what that means now is that I can choose, you can see that now there's a drop-down menu appearing. I can choose within these different combinations of what I'm looking at. And so let's say we want to look at just interferon gamma total, like a similar readout to ELISpot. So you can press on that, press OK, and then you can see that the spot counts get adjusted for that readout. So this is the way to filter. And of course, maybe, if you're interested in grams and if in total numbers only, you can always just go like that and copy it somewhere else. But there are ways of building this table a little more extensively. So for example, we could really, be interested in all of these channels, but we can change the LED filter with the stimuli information, for example. So I'm going to flip. I'm going to put the stimuli in on the filters and a filter on the rows. I basically changed the organization of it. And then we can filter on, let's say, SARS-CoV-2. So I'm interested in the SARS-CoV-2 response. And now I'm getting the spot counts for just the SARS-CoV-2 response for all donors for all of these combinations. And you can, again, here also choose what you're looking at. For example, maybe we just want to look at granzyme B total, interferon gamma total, and TNF total. So you can filter out the information that you're interested in like that as well. It's a very similar process to ELISpot, like what I showed with the pivot tables in ELISpot, but with FluoroSpot, there's just this aspect of LED filters and being mindful that you need to kind of specify which LED filter you're looking at. So yeah, these are very flexible tables, and we can always spend a bit more time one-on-one with any of you and go through this analysis. But it just takes a little bit of playing around with this to figure out how to utilize this fully. And the last thing I want to show you before we take some questions, I'm just going to close all of these, is that you are also able to, with our Apex, it is possible to export single well information. So now we have mostly worked on aggregated data tables from the single well. So average spot counts per well, but we haven't been looking at the single spots themselves. So each spot, of course, has its own associated value. So, if you click on any of these spots, you will see that they have intensity, diameter, and spot volume values. So these values, they're not readily available in the plate information because we don't export them automatically. But what you can do is you can export them yourself. So let's say you're interested in several wells and their and the information about each spot within those wells. All you need to do is very simple. You just highlight the wells that you're interested in. So let's say we pick these 2 wells. You just highlight them with your mouse and then right click. Highlight them with the mouse and then go into the export selected wells data Excel. And then I'm going to save that onto the desktop. And let me open that. So here we are, plate 2 wells. And that's the Excel from those 2 wells. So I'm just going to open it. And again, another table that we're looking at, which is very, very big. But as I say, it's very easy to deal with because we have this pivot function integration ready. But if you look at this table, we have several rows of plate 2 from well C8. And you can see that the spot is indicated as 1, 2, 3, 4, 5, 6, 7, 8. And that's because the well that we were looking at had 8 spots. So basically you're looking at those 8 spots in these first few rows. And then the rest of the rows are from the other well that had way more spots. This one had 120 spots. So you basically have exported all of this information about every single spot in the well. And the information that you're actually getting is spot position, which is usually not that useful, really. But the useful information would be the relative spot volume for that spot, the diameter, which is the size, basically, of the spot, and also the spot center intensity. So if you want to get this information into, again, an aggregated table, it's a similar principle. You can highlight this full table with Control A. So I do that. And then we go Insert, Pivot Table. And then we can basically build in similar concept again, like the pivot tables like before. We just build our table as we wish. So if we want our well information under the columns, so we have C8 and C9, and then we can move the spots under the rows. And then we also have, let's say, information we're interested in is the relative spot volume. So we can move that to the values. And here we are. Now we end up with 2 columns that are C8, those 8 spots that we had in this well, and then all the other wells and their corresponding relative spot volume. So now you can take this information and do all sorts of analysis that you might be interested in. And I can show you the example of how this information can be used is, for example, if you're comparing the relative spot volume between 2 different wells, what you could do is, let me just go to an example. So in this case, for example, if you're comparing the relative spot volume for each spot in peptide 1 and peptide 2, even though they have similar numbers of spots, they might have different secretion levels. And you can see that peptide 2 has a few larger, higher secretor cells. So if you were to gonna plot this information that I just showed you in the table side by side, you can analyze if peptide 2 or peptide 1 is producing more of the analyte or more of the cytokine than another peptide. The important thing to remember is that RSV values can be affected by the development time of a substrate. So the longer you develop, the stronger the spots. So, the reproducibility between the plates is important to stick to the same development time. But the good alternative would be to go for FluoroSpot, which doesn't have that issue of overdevelopment. And the RSV values are very robust because they can't be affected by assay conditions. But this is just the kind of thing to keep in mind. So with this, I think I will end now, and maybe we'll have a few questions. So yeah, thanks for listening. I hope it was somewhat clear and not too fast. Thanks.
No, I think the presentation speed was perfect. If there were any questions that anybody had, obviously we were going to be able to capture them in the chat. So, but with regard to questions, let's go ahead and start with, is the Apex considered validated software, specifically 21 CFR Part 11?
Yes, it is.
Yes.
Excellent.
Yes,
And
it
I
is.
think in
That
part-
was an easy one, Jason. Thank you.
Hey,
Yeah.
I mean, it's an important one though, if I understand, because as a follow-on to that, there was also some question about what's the audit trail functionality
Yeah.
of Apex software?
Yeah, so the really good, great question about the compliance. Yes, there are many, regulatory requirements for softwares like ours, and we are meeting the compliance really well. And we continuously make changes or improvements in that field. But yeah, our readers are used in regulatory environments quite frequently. So several things with the software that are important to remember is the audit trail, like what Jason mentioned, and we can show you that in just a second. But there's also, ability to restrict users to do certain things on the software, give different permissions, have, the audit trail that we will show you, data integrity. So all of these things. And we have, maybe Berend or Jason could actually share it in the chat. I'm thinking we have a very nice PDF that could be a resource to read about how we are compliant with CFR Part 21, Part 11. But while we're doing that, I can show you the audit trail. So I'll just share my screen again. So now I'm just looking at the plate that I have read. So there was plate 2. And with every plate with Apex 2, what we have implemented, especially with Apex 2 version, is a history table or a history PDF. Basically, this PDF is non-editable PDF that has information about each session, each login, whoever done anything to the play and the data. So right now you can see that it was just a session user, when I started using it, when I stopped using it, who was logged in, and also what kind of information we've had at the time of saving. So we have the area of interest information, spot counts, RSV values, and everything like that. So that is everything basically that is done to the data is logged in that history PDF. But we also, in the plate information itself, we have a plate history tab over here. So if you press on the plate history tab and select the session that we're interested in, we can look at that information in a little more nicer way. So for example, in the spot forming units under this session, we have these spot counts. And if someone goes in, makes changes, saves the data again, you're gonna have that logged under the sessions. So this is the audit trail that comes with Apex 2.
Excellent. I think in kind of the same vein, I've got a question. Is the software compatible with Windows sign-on? So basically using your Windows credentials to log into Apex.
So currently it is not. It is a frequently requested feature in the past few months. So we have taken it up with our software development team, and it is something that we might be implementing sometime in the future. So yeah.
Excellent. And does the software offer any APIs?
Yeah, so API is like a way for software to talk with other software. And specifically in Apex version 2.0, we implemented an API for the software to interact, for example, with robotic arms to install automation with your Iris 2 or Aster 2.
Yes, I think it's important to mention the Aster 2 and the Iris 2 have had modifications to allow the robot arm to place the plate into the receiver, whereas Aster 1 or Iris 1, that functionality is different. So if that is important, we have that ability to maybe potentially talk about updating that either with a trade-in or a different way for your laboratory. We received a question just recently. How do you edit the size and intensity parameters for FluoroSpot? And can you speak to some best practice principles around that, since it can modify your output?
Best question. It's the most important question in the method, I would say. And it comes up a lot, and it's a very, very important one because, as you say, it does affect the output. So I have a FluoroSpot plate open so I can show, and I think that, you know, we can have a discussion with Berend about this, because it really is you know, it's so far it's a subjective decision that is based on the researcher's needs, but it always needs to be done in consideration of your controls and your experimental conditions. So for example, if we're looking at interferon gamma response right now, we are looking at different stimuli. So we have unstimulated wells, SARS-CoV-2, and CMV stimulated wells. Now, important thing is that in unstimulated wells, even though you have cells that haven't received any specific stimulus, they just have media added, there is going to be some spontaneous secretion of interferon gamma. So if I click on that one of these wells, you will see that there are some spots. Now, these spots can be biologically real, and they will have, of course, their own size and intensity values, and they can be quite large sometimes. So the goal is never really to fully exclude spots from your unstimulated controls, because they can be biological. So if you go and look now into our peptide stimulated controls, we obviously see an increase in the number of spots. But as you can see, there are some strong spots in your controls too. So the way that you can decide, basically the decision is very subjective, and each experiment that you do and stimulation conditions and the media that you will use will probably affect the fluorescence intensity, the secretion levels, and so on. So you will need to adjust the size and intensity values depending on your setup. Our defaults are good, but the way that you would do it is basically by going between the unstimulated controls and the stimulated conditions and deciding where is that fine balance without picking up too much background. Because if I, for example, decrease the intensity threshold to 10 now, you can see that I start getting more and more spots, but I'm starting to pick up membrane background. I'm starting to pick up very, very faint stuff. And of course, that's not really real, right? So finding that value isn't straightforward, and there isn't really a consensus on how to do it in like a mathematical way, but it's more of a subjective kind of decision going between your stimulation and unstimulated controls. Berend, do you agree? Do you have any input on this approach?
No, so as you say, it's quite subjective, and everyone has their own methods of doing it. I think it's the way you showcase it by comparing your unstimulated conditions with your peptide conditions or stimulation conditions, and verifying that you're really picking up the spots is a good way to do it. I can also mention that there is a great publication, the name slips my mind right now, in which they really talk about this in ELISpot, but I think it's quite applicable to FluoroSpot as well, really about the process of how, not necessarily how do I set my parameters, but what is really the method and practice of going through setting changes and applying that to my whole plate, for example. I can look around in a bit and see if I can find a publication, and I can share it later.
Yeah, no, that would be great. But yeah, it's not a straightforward approach, and it's analyte dependent. So now we've looked at interferon gamma, but granzyme B and TNF have much higher spontaneous secretion and can give you 100 spots in your unstimulated controls, while in your stimulation you might have 200. So it's a fine balance, but it's not so straightforward. Yeah.
Follow on for that question. If we save the template, does it save the size and intensity parameters as well, or just the layout?
Yes. It does.
So it saves all of the parameters.
Yes, exactly.
So it is a great way of, you set up a template for a study that you would be running where, you will have a very typical plate layout and you want to keep the same parameters. I think that's the best use case for a template. Really, you set up those parameters in a case study in the first plate, and then you just use those settings and that, plate layout for the rest of all your plates. And you don't need to think too much about changing any settings, and just really being secure that, the settings that are applied in the very first plate are the exact same ones that are applied in the very last plate that you will be reading out.
Right. Excellent. Looks like the questions have stopped. If there are any more, obviously, you're more than welcome to contact any of us. I do want to say thank you very much for your time and attention during this training session. If there are additional bits of information that you do want to learn about, I'm more than happy to gather new topics so that we can hold additional training sessions for you. And also, Renata is our North American field application specialist, so she is also available to work with you on an individual basis to make sure that the data that you're getting out of all of your ELISpot and FluoroSpot assays are going to work for you the best that they can. On behalf of BabTech, Berend, Renata, myself especially, thank you so much for your time. We really appreciate you attending. Enjoy the rest of your week.
Thank
Thank
you,
you all.
everyone.
Bye.
Bye. Take care.